Good morning, it's June 9th and this is your daily brief in science. Here's everything you need to know. A new set of trial results on Survodutide is sharpening the potential for MASLD therapies as synchronized MASLD met its co-primary endpoints over 48 weeks, with the majority seeing meaningful liver fat reduction and a notable portion normalizing liver fat. Across the same program, Boehringer Ingelheim's Survodutide showed strong fat loss effects, including large reductions in visceral fat and liver fat, while lean mass loss stayed limited at the highest dose, indicating fat-driven weight loss and improved metabolic health. In Synchronize 1, 725 adults without type 2 diabetes were randomized to weekly 3.6 mg or 6.0 mg doses versus placebo for 76 weeks. With primary endpoints focusing on percentage weight change and a high threshold for achieving significant weight loss. Survodutide remains investigational and is not approved, carrying regulatory designations such as FDA Breakthrough Therapy and Fast Track, EMA Prime, and Breakthrough designations in China and Taiwan, signaling potential expedited review pathways. Global Phase 3 programs, LIVERAGE and LIVERAGE-cirrhosis, are evaluating Survodutide in MASLD-related fibrosis at advanced stages, signaling broader trials in MASLD-related liver disease. The drug is licensed from EurekAlert Pharma to Boehringer Ingelheim, which leads global development and commercialization. Zealand stands to royalties and milestone payments totaling up to mid to high single digits and up to Euro 315 million. Positive Phase 3 results from Synchronize 1 and Synchronize MASLD were presented at major conferences and published in NEJM and Nature Medicine, underscoring robust data for Survodutide. The releases align MASLD/MASH epidemiology with the growing need for treatments that address visceral and liver fat and overall metabolic health. Safety aligns with GLP-1 based therapies, with gastrointestinal events commonly reported and discontinuation around 19% at higher doses, but no new safety signals. Beyond weight loss, Survodutide aims to improve metabolic health by addressing visceral and liver fat, inflammation, and fibrosis, with outlines for future trials and global development plans. The program combines GLP-1-mediated appetite suppression with glucagon-driven hepatic fat reduction, aiming to treat obesity-related metabolic dysfunction and liver fat accumulation across broader populations. The message reiterates that Survodutide is investigational, with safety profiles consistent with GLP-1 therapies and similar discontinuation rates at higher doses. A complete central nervous system connectomy of an adult fruit fly has been mapped, linking the brain to the nerve cord to trace information flow from sensation to action across the organism. The map covers all neuronal connections at the synapse level and is freely available online for researchers to study brain-body interactions and behavior as a whole. This work extends prior brain-only maps by including the nerve cord, enabling hypotheses about motor control and behavior to be tested in a holistic brain-body context. The project is supported by NIH, NSF, the Brain Initiative, and other funders, with several authors and institutions disclosing financial interests and patent considerations. The data offer potential implications for artificial intelligence and robotics by illustrating how decentralized embodied networks coordinate actions and navigate environments. Future directions include adding neuropeptide data to the connectomy, expanding to more complex organisms, and exploring whether distributed control is conserved across species, including possible implications for human neuroscience and AI design. The dataset is open source and freely available online, funded by the Brain Initiative, NIH, and NSF, intended as a benchmark for computational neuroscience and mammalian comparisons, akin to the Human Genome Project. The study, titled Distributed Control Circuits Across a Brain and Cord Connectomy, was published in Nature on June 8, 2026. A key finding is that many behaviors are controlled by local, body-part-specific neural circuits rather than a single centralized brain hub, indicating distributed control of movement and action. Representative findings show motor control is largely local to each limb, with interconnections coordinating complex movements like walking, while wings and other appendages participate with feedback from sensory and endocrine systems. Methodology involved thousands of serial ultra-thin sections imaged by electron microscopy, with AI-assisted alignment to create a high-resolution synapse-level wiring diagram, now publicly available as the BANC-FLYWIRE dataset. The study describes a multi-step process of preparing serial sections, imaging with electron microscopy, and using AI to assemble a cohesive three-dimensional map linking brain and body circuits through identifiable neurons. A new Octopus-inspired soft robotic arm from the Italian Institute of Technology embeds tactile sensing directly into its suction cups, enabling contact detection, force estimation, and autonomous grasping in air and underwater. The effort, led by Barbara Mazzolai and her bio-inspired soft robotics unit, advances tendon cable optimization models and 3D printed soft endoskeletons to link perception and action inside the arm itself. Building on IIT's broader Octopus robotics program, the work uses computational tools for optimal cable arrangements and manufacturable endoskeleton pathways to support modular sensing. Potential applications include handling fragile objects and biological samples underwater, as well as inspection and maintenance tasks in hostile industrial and natural environments. The design is modular, with adjustable suction cup counts and placements to fit varied tasks, supporting marine biology research, subsea maintenance, and other underwater operations. The system's modular and scalable nature allows tailored grasp distribution and enhanced environmental perception for specific missions. This research has broad implications for autonomous underwater vehicles and robotic manipulators, enabling precise, context-aware interactions in delicate or hazardous ecosystems and paving the way for multi-limbed platforms with distributed sensing. The approach mirrors the Octopus's decentralized nervous system, enabling rapid autonomous grip adjustments in challenging underwater settings and improved handling of delicate objects. Lead author Emanuela Del Dottore emphasizes embedding sensing into the robot's interface with the environment, signaling a move toward embodied autonomous systems with immediate motor responses to tactile input. Future goals include increasing payload capacity, expanding grasp versatility to more underwater objects, and extending the approach to more complex environments for fully autonomous soft robotics. Suction cups use silicone cups with miniaturized optical tactile sensors that detect changes in reflected light to estimate contact force and direction, enabling distributed local sensing and peripheral control. The arm's miniaturized optical sensors inside suction cups measure force magnitude and direction, as deformation alters LED reflections, coordinated by a distributed control system. A Nature study from year 2026 reports a CRISPR-Cas12a2-based method that uses RNA guidance to target cancer-specific transcripts, focusing on mutant p53 to trigger rapid chromatin shredding and kill cancer cells while sparing normal tissue. Background shows researchers are aiming to improve delivery to tumors and to test across brain, prostate, and ovarian cancers through ongoing collaborations as they pursue scalable genetic medicines that could address multiple cancer mutations. The work points toward RNA-guided, customizable specificity that bypasses the need for druggable protein pockets and could apply to roughly 40 to 50% of human tumors, with proof of concept experiments in cell lines and tumor organoids showing selective elimination of mutant p53 transcripts and a therapeutic window. Correspondence author Jennifer A. Doudna is listed for study communications, and the article provides extensive supplementary materials including uncropped gels, gRNA and oligo catalogs, substrates, cell lines, plasmids, and a peer review file. Ongoing challenges include delivering Cas12a2 and guide RNAs in vivo, with exploration of viral vectors, lipid nanoparticles, and other delivery methods to reach tumors effectively. The approach integrates RNA recognition with chromatin-level genome manipulation to selectively disrupt cancer-associated genetic alterations, signaling a shift toward using RNA footprints to provoke genome-wide chromatin destruction as a therapeutic strategy. Development continues to broaden testing to additional cancer types and to refine delivery, while researchers explore potential applications beyond cancer, including targeting virus-infected or aging-affected cells. The study, published by Nature on June 8, year 2026, follows a timeline of submission in early November year 2025 and acceptance in late May year 2026, reflecting rapid progress in this area. Overall, the research emphasizes moving away from protein-targeted strategies toward exploiting RNA signatures that trigger chromatin-based cancer cell destruction, with a focus on selective targeting and scalability. A new multi-institutional study maps how neural circuits traverse from the brain into the spine, offering a data-driven view of connectivity across a brain-spine connectomy. The work, carried out by the BANC-Flywire Consortium, leverages extensive datasets and advanced analyses to chart distributed control circuits that span the brain and nervous system. Across a broad author list with equal contributions from Bates, Phelps, Kim, and Yang, the paper presents a comprehensive approach to neuron metadata and cross-dataset alignment. Published in Nature on June 8, 2026, the study is titled Distributed Control Circuits Across a Brain and Cord Connectomy and is supported by a formal D01 indicating its publication status. The research emphasizes a connectomy framework that integrates brain and cord structures to reveal how neural circuits are organized across levels of the nervous system. Supplementary information is substantial, detailing neuron metadata from datasets such as FAFB, MANC, and male CNS, and employing analyses including PCA, UMAP, spectral clustering, and functional annotations. The material describes cross-dataset neuron metadata alignment, UMAP embeddings, clustering results, and data quality artifacts bounding boxes, signaling a thorough cross-dataset approach to neuron types and connectivity. Corresponding authors are based at Harvard Medical School and affiliated institutions, underscoring a broad, multi-institutional collaboration. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.